Genesis. So this is going to follow on from that podcast, The Primitive Streak. And if you haven't listened to that already, then go back and listen to that. The link is on Blackboard. What I'm trying to do with this one is take you a little further inside and show you how the baby actually organizes itself in the very earliest stages after fertilization. So let's go. And this is from the paper by the group that they were talking to in that podcast. So this is the science that is based on. That's like a more of a general explainer for everybody. And then, once you've got used to that, then we're ready to go into this. This is our next level. So here we go. All right. So the primary tissues of any kind of embryo, mouse or human, any kind of mammalian embryo anyway, are going to be the Trophectoderm and Epiblast and the primitive endoderm. And they're all going to be established by day 4.5 in a mouse. And I think it's a little later in humans. Anyway, this is the way that it works. So you're going to start out with one cell, and that's going to divide to make 2, it's going to divide to make 4, and it's going to divide to make 8. All right, and at this point, all 8 of those cells will actually be equivalent. And they're going to be expressing 2 different genes. One is called CDX2, and the other one is called OCT3 or OCT4. And so, CDX2 is the caudal type, so like for the tail, homeobox protein number 2. So that's what it is. And homeobox proteins are involved in patterning. So these determine, in part, where your different body parts will end up in the finished adult. And so they switch on and switch off at different times during growth. And they are going to direct growth to make sure that you end up with the right number of arms and legs or, whatever you have, depending what kind of animal it is. All right. So at the 8-cell stage, you've got all 8 cells, and they're expressing both CDX2 and OCT3 or OCT3/4. We know that all the cells are equivalent at this stage because you can actually do some tests. So in some cases, there would be groups of people that have a high incidence of genetic problems in their babies. For example, the Ashkenazi Jewish community, I know, gets a high rate of Tay-Sachs. And that's a thing that you can test for genetically. And they do that by actually taking out one of the cells at the 8-cell stage. So they'll take an embryo, grow it up to the 8-cell stage in a laboratory, and they will then take out one of those cells and do the genetic tests on that one cell. Read out the genes, see whether or not they have the mutations that are going to cause problems later on. And you know what? You take out 1 out of 8 cells and you don't get 7/8 of a baby because none of those 8 cells is actually committed to be any part of the baby yet, which is pretty cool. Okay, next comes the 16-cell stage. And in a mouse, this is going to be at about 3 days after fertilization. And so at this stage, the cells make a choice. So at the 8-cell stage, they were expressing both CDX2 and OCT3 or 4. Now they're going to switch, and about half of them will express CDX2 only, and half of them will express OCT3/4 only. And so there aren't going to be any cells that are expressing both anymore. They all make a decision, and you're going to see that a couple times throughout this early process. So they make their decision right here. Each cell just randomly commits, and so half of them will randomly commit to one thing or the other. And it works because you end up with about, statistically, you end up with about half the cells going one way and half going the other. And then once these cells have committed, they are going to reorganize. So it's going to be commit and then reorganize. And so all the cells that happen to make CDX2 first are going to form themselves into a ring around the outside. And all of the cells that decide to express OCT3/4, just randomly, because that happened to work, are going to line up on the inside. And they're going to divide a few more times until you get just the right number of cells to make up each of these layers. Then the outermost cells, these guys are committed. So they are going to form the Trophectoderm. This is going to be something like the eggshell, basically, if you imagine a human baby as a baby chicken. Let's see. The embryo is actually going to come from the stuff that's inside. So the thing that's the inner cell mass, that's the ICM right over here. And so at the next step, those cells of the inner cell mass, which we're expressing OCT3/4, are going to make another decision. They're going to commit once again. And so they're going to commit to either express GATA6 or express NANOG. We've got sequences of NANOG, interestingly, going back to Neanderthal humans. These are cavemen. And there are some little differences between human and human ancestor NANOG and some of the other ones that we find in, for example, chimpanzees, other kinds of apes. Anyway, once they commit here, they're going to do the same thing. And so they're going to reorganize themselves again. And all the ones that ended up making GATA6 are going to line up here, and they're going to form the primitive endoderm. The rest that decided to make NANOG, this is the marker for the cells that are going to form the epiblast. And as you probably remember from the podcast, the epiblast is going to form all the cells of the baby. So it looks like the primitive endoderm, you'd say, oh, that's probably going to make the endoderm of the baby, but it's actually not. The primitive endoderm is marking a place. So it's marking the site where the endoderm, the definitive endoderm, as they call it, is going to form. And that's going to be a little later. That's going to be something we pick up from the videos that are online. The epiblast is going to make all the cells of the baby, and some of those cells from the epiblast will go out and they will replace the primitive endoderm and make the definitive endoderm later. And then the trophectoderm, again, it's all just cells, and they're the cells that are genetically identical to the ones in the baby. They are just not expressing all of the right genes that they need to be part of the epiblast. And it's good. You need cells that are doing each of these things. So weirdly, yeah, only some of the cells in this original fertilized embryo are going to go on to be the baby. And those cells are not going to be set until you get to the stage where you actually have an epiblast, and it's organized like that, and where you actually get NANOG expression. And so that's going to take place about 4.5 days after fertilization for the embryo of a mouse, and it's going to take place a little later for the embryo of a person, as they discussed in the podcast. So let's actually go and see the human data. This is just like a little sort of a general thing to show you what's going on and to explain how the expression of genes changes in the early baby. And these are a couple of the genes that change. These are the ones that seem to be the big markers, like the big differences. But there are many, many, many other genetic changes that happen around this time. And let's go to full mode. There we go. With subtitles, which I should have put on the other one. Oh, well. Okay, so here are some of those human embryos that they were talking about in the podcast. And so these are human embryos that are going to be cultured in different conditions. Now, full-on air gives you about 21% oxygen. That's just like the amount of oxygen that's in the world around us. 5% oxygen is going to be a little closer to what you would expect to find inside of the mother's womb. So that's going to be a little more normal. And as you can see here, so let's go through these different stains. We've got DAPI. DAPI is a blue dye that sticks to DNA. And so this is just going to show you where the nucleus is of each cell. So you can't see the rest of the cytoplasm of each cell, but you can see the little circles, and each one is a nucleus of a cell. Then we've got OCT4 right here, and so the human OCT4, and so there we go. And so OCT4 is going to be expressed. These are the same picture anyway, and so when you see things in different areas, that means it's different cells that are expressing. So the little white box here is showing you the extent of the OCT4 expressing cells. And so you'll remember the OCT4 expressing cells are going to be in this area. So what we're talking about is this inner cell mass right there. And so you can see a little baby human inner cell mass. Now, in the experiments that they did, they were actually, so the cells are actually in sort of a growth medium. It's kind of like an energy drink. It has lots of amino acids and sugar and things that baby cells need in order to grow. And so it's not actually at 21% oxygen. It is however much oxygen dissolves into that fluid from straight up air. And so these guys down here are really going to be hypoxic, basically. These are babies that don't have perhaps quite as much oxygen as they would in real life. So anyway, there's OCT4 and showing you the inner cell mass, and that's kind of neat. And over here we have PKC, which is protein kinase C. That's something that we talk about, I believe, in a later lecture when we get to substance P. But protein kinase C is going to be a kinase, so it's going to add phosphate. And this one is a very common kinase that has a lot of different targets. So it adds phosphate to a lot of different things. It's very important in developing cells. And so as you can see here, it's going to be a marker of the Trophectoderm, most likely. It's showing up around the outside. F-actin, filamentous actin, is going to be also in rapidly dividing cells and cells that have sort of a stronger cytoskeleton. And so you can see, once again, there's a lot of F-actin that's forming around the edge. And so again, yeah, this is our equivalent of the eggshell that our little baby's going to grow in. And so there's a complete little human embryo with a little blob in here being the thing that's going to turn into a baby. And yeah, if you deprive the little embryo of oxygen, then you get one of these malformed lumps down here. And yeah, you've got fragmented nuclei, little broken bits of blue. Basically, this is a baby that's dying because it's not getting enough oxygen at the same growth stage. Okay, so it takes a lot of oxygen to keep the baby going. And shown here, once again, we've got, so this is day 5 and 6, little blastocysts, which is one of the stages of development, where we have the 2 layers. And so the DAPI shows you where the nuclei are of all the cells, so there and there. And these are all going to be in the high oxygen, the 21% oxygen sort of environment, so that they grow properly. And this is going to be equivalent to what would happen with a functioning circulatory system. So the mother's circulation is actually going to deliver the oxygen to keep these embryos fully oxygenated, and so they're going to grow just like this. Then you can see GATA6 here, and you can see OCT4. And OCT4, just like we said, is going to light up the middle in particular. GATA6 is going to light up the outside. And at this stage, you can see there are still a few cells that are trying to express both of these things. And you can see them coming up over here as either green or yellow. And these are on their way. They're going to migrate out, or they're going to switch off the GATA6 and just make the OCT4. And these are both just transcription factors that'll go and do a little bit of patterning in other parts of the body. So GATA6 is going to be a marker of the first layer, so that's going to be equivalent to what we were describing here as CDX2. GATA6 is going to play the equivalent role. It's going to be also turned on in those cells that are making the shell effectively. All right. Yeah. And so here are little human cells, and they're developing all the way out, day 7 to 8, 8 to 9, 9 to 10, and 10 to 11. And these are going to show expression of some of the different things. So here's our F-actin, which is marking any cells that are in the process of moving, for example, migrating cells. And it's also going to be a good marker for the outer shell. And then we've got our protein kinase C right here. And then in the center, we've got DAPI, so that's all the nuclei, and OCT4 and protein kinase C and F-actin. And you can see those OCT4 expressing cells are going to be this nice sort of whitish purple color. And those are right in the center. These are going to be the actual epiblast cells. So they keep their OCT4 expression. They're also going to turn on nanog expression, like we showed in that first slide. And yeah, nanog plus OCT4 means epiblast. When you get both of those things on, that marks a cell that is going to go on to actually form some part of the body of the baby. Now at this stage, you can still mess around with these cells. You could still take a couple of these cells out, and you would still get a full baby, because each of these cells is a stem cell. Each of these is a cell that could turn into literally any tissue, be it brain or, left foot. And so, yeah, they have a lot of potential. They can go on to make any part of the baby, and they have not yet committed to do any particular thing. And so you've got your, these cells are going to organize, and they're going to make a yolk sac, which is going to be something like food. But again, it's part of the baby embryo that's going to be sacrificed as food. And you've also got the cells that are going to go and divide and actually become the baby. Yeah. So here you go. You can see here the actual sort of structure of the epiblast in these cells. And yeah, so let's see. We start out down here with the cells that are expressing either GATA6 or OCT4, so they're going to be the little pink cells. And these are the ones that are going to eventually become the epiblast, but they haven't organized yet. They haven't committed fully yet. And so you're going to have cells that make up the sort of sac around the embryo. And the embryo is actually going to hatch out of this and then implant itself in the mother because it needs to get more food. The embryo can survive on the food that it's brought with it, the food that was in that original eggshell, for a certain amount of time. But then after this stage, they really need a bigger source of, just sugar and oxygen and everything to keep them going. And so that's what they're going to do. They're going to burrow in and try and find some veins or arteries from the mother and actually merge with those so that they can get a better food supply and oxygen supply. And so that's what a little baby human looks like. And these are the stages that we did not know about before these experiments were done, because we had the early stages back here around day 5, but that was pretty much all we knew. So all the new stuff from this paper is what's going on here at day 6, 7, 8, 9, 10, and up to 11. And then that's where these embryos seem to start to go a little bit different. They no longer resemble true human embryos. We think that's the point at which you start to need some sort of signal from the mother in order to keep the embryo developing on the normal path. It'll kind of grow itself up to 11 days, but after that, yeah, it's going to need a little bit of help and a whole lot of feeding. And so that's where parents come in, right? Taking care of the baby. Okay, so one other little topic on the end of this, and this is going to come back when we get to the neurobiology week, which is coming up. So there are chemicals in your body, and in this case, they're actually small proteins. These things are called neuropeptides. And so there you can see the word written out, neuropeptides. And oxytocin is one example of a neuropeptide. Oxytocin, shown here, is a neuropeptide made of these amino acids, and so they've written out the amino acids with 3 letter codes, but that's going to be cysteine, tyrosine, isoleucine, glutamine, aspartic acid, cysteine again, and remember cysteines have a sulfur on the end of it, and they can form this disulfide bridge. The 2 sulfides link. Then there's a proline and a leucine and a glycine. That together is oxytocin, and that will actually bind to receptors, and it alters our mind. So oxytocin is given to women both to induce labor, so it forces the baby to come along, and it also is thought to create or just help sort of maintain feelings of love and attachment. And so, part of the reason why people love their babies is this little neuropeptide, oxytocin. It's weird to think of a little chemical that's actually coming from inside your body as modifying the mind, but that is exactly what neuropeptides are, and that is exactly what we think they do. There's a pretty rich literature about some of these things, in particular oxytocin. And so yeah, when we get to Neuropeptides Week, maybe that's one that you want to research a little more. I don't know. But there are lots of these neuropeptides. One of these is called metencephalin. I think I stole this from a Spanish language or Italian language site. And so that is going to be one of the endorphins. And so again, endorphins are a thing that you make in your mind. It's a little tiny peptide, neuropeptide, in fact. And it's going to bind to its own receptor. The receptor is going to be one of those G protein coupled receptors because, yeah, there are a lot of them. They're involved in most every kind of signaling that happens in the body. And interestingly, morphine over here, so they've written it as morphina, but it is morphine, actually works by mimicking that. So it's going to bind to the same receptor as a metenkephalin. And you can see the part of the molecule highlighted in red here, which is going to match the part of the molecule that highlighted in red there. And you see they have a very similar molecular structure. And so they do bind to the same protein in roughly the same way. And so morphine is a substitute for your natural metenkephalin neuropeptide. So yeah, you can look at this as you can make your own morphine, or you can look at this as you can inject, and please don't, but you can inject something that mimics a neuropeptide in your own mind. So it's pretty cool either way, right? And there are a lot of these neuropeptides, so I just want to introduce those right now so that you can get to know them, but they are very interesting, I think. And so we have lots of these things. Neuropeptide Y is a famous one. Galanin's a famous one. Leptins, oh my gosh, anytime you feel hungry, think of leptins. Neurostatin, yeah, cocaine and amphetamine regulated transcript. Interesting ones. So these are some of the diseases that are associated with genetic changes. So there are people and animals that have single nucleotide polymorphisms. So single nucleotide means one nucleotide, and polymorphism is basically a change or a difference between two people. So single nucleotide differences in these genes are associated with, for example, for leptin, obesity. So leptin is a thing that controls hunger. And if you are lacking a functional hunger control and you have plenty of access to delicious, food, we live in the land of barbecue, then yeah, obesity is certainly something that could happen. But you've also got some of these that are involved in, for example, anorexia or different addiction behaviors. Yeah, and also even in arthritis. And so there are also things that will bind to these same receptors as the neuropeptides and either enhance their effect or block their effect. And so this is a lot of medicinal chemistry. This is what you get paid the big bucks for when you're working at a big biopharma company. These are going to be the drugs that make you feel better or feel worse or alleviate anxiety or depression. They're all going to work through these little neuropeptide receptors, and they're going to work by mimicking one of the neuropeptides that we have in some way. So I think that's just a very interesting area of biology. And yeah, if you want to continue in biology and make a whole lot of money, then you want to get really good at your neuropeptides. Understand some different ones, get a good basic idea. So there you go. It's an interesting topic, and I wanted to make sure that it was presented to you so that you could see it and so that you could know about these things. So I think that's the end of this section. Yep, that's it. All right, and thank you very much. Let's end this show.